Chapter 21. So in the interest of timing, I might be skipping some of the slides just so you're aware, but I'll make sure that I tell you what you need to know and what you don't need to worry about. Okay, so the heart is a pump that provides major force, causing blood to circulate in the blood vessels. That is true. We learned that in Chapter 20. But the highways that carry that blood are going to be your arteries, your veins, your capillaries. So we have two different types of vessels, just like we had two different types of pumps. Let me change that color. Just like we had two different types of pumps in the heart. We have two major types of vessels. We have the systemic vessels being the ones that go from your left ventricle out to your body and then your body back to your right atrium. And then we've got the pulmonary vessels, which are going to be leaving the right ventricle, going to the lungs, and then from the lungs back to the left atrium. So just like we had two pumps in the heart, we basically have two highways, one that goes to the lungs back to the heart, one that goes to the body back to the heart. The functions of peripheral circulation. I'm sorry, my allergies are bothering me, so you're going to have to put up with my runny nose this morning. One, it carries the blood. It takes the blood on a round trip from the heart to the tissues and back. Ultimately, whether that's the lung tissue or the body tissue doesn't matter. The fact that we've got a highway that does that is the major function. Exchanges of nutrients, waste products, and gases. Okay, so we pick up nutrients in one place, we drop them off at the cells of the body. We pick up, well, at one place, the intestines normally. We pick up oxygen at the lungs, we drop it off at the body. But at the same time, you also have to remember that the cells of the body are living things, which means that they contribute. Really? They make waste, which means that also has to be picked up to be dropped off. Whether that's carbon dioxide that's picked up and dropped off at the lungs when we breathe out and get rid of it, or whether that's waste products that have to be taken to the kidneys to get filtered. It transports substances. We're not only talking about the nutrients and waste that it's transporting, but things that get made in one part of the body and then need to be placed in another part. We talked about the posterior pituitary creating oxytocin, the thing that causes uterine contractions during labor. Well, that actually has to go into the body or into the blood, and then the blood has to transport it from up at the posterior pituitary in the brain down to the secondary location, which is the uterus for uterine contractions. It helps to regulate the blood pressure. So our blood vessels actually have smooth muscle, excuse me, that wraps around it kind of like a drawstring. And what that means is I can have big, huge arteries, or if I contract those muscles down, I can change their diameter. I can make them smaller, meaning that I can change the blood pressure. Even if I've got large amounts or small amounts, I can change the size of that vessel so that my blood pressure, which of course is how hard that blood is pushing against these walls, stays maintained. And we'll talk about why that's important in just a bit. And it can direct blood flow to tissues. It can flow with purpose, directing circulation to areas in need. For example, right now I'm doing a lecture. I don't really need a whole bunch of blood going to my skeletal muscle like I'm running a marathon. But if that changes, if I go to Zumba class, or if I have, I don't know, the mountain lion showing up in my office where I'm at, then yeah, it may change. So I can actually change where my blood is flowing to. Okay, so the homework that you were assigned, that actually covers the sections that we're skipping here. We started at section 1 in the chapter, now we're going to section 6. So just like water flowing through pipes, certain relationships affect the function of the cardiovascular system. These include things like your blood pressure, your blood flow, the resistance to that flow, and the control mechanisms of blood pressure and blood flow. So, laminar flow. I'm going to change colors real quick. I'm going to go with green, light green. So laminar flow. Laminar flow is the movement of fluid into or through a smooth tube. And it is actually very streamlined. If you look over here, you'll notice that the blood flowing through this tube is actually kind of in a bullseye shape. Now I want to explain this to you. It's in concentric layers. So the speed of this blood is in concentric layers. Now, the outermost layer out here, this layer right here, is actually the slowest moving layer of the blood. You've got to remember that this blood is right next to this wall of this tube or artery or vein or whatever, and that means that there's drag on it. The tube itself is completely stationary, meaning that it's not moving. So as this blood flows through here, as it actually travels down this tube, it's getting dragged on and slowed down by the fact that it's right next to the stationary wall. Now, if you look, there's a second layer right here. Okay, now I'm going to erase this so you can see it again. That second layer in is right next to the slowest moving layer of blood. So it's actually slightly slow. Not as slow as the one that's right next to the wall, but it is slow. Same with the next layer in. The fastest layer is this layer dead center in the middle that's next to the second to the fastest moving layer of blood. That concentric layer, I guess, layout is how it becomes fastest dead center in the middle here. Okay? Now, turbulent flow. Everybody's probably heard of turbulence on an airplane. So turbulent flow is when the flow exceeds a critical velocity, when fluid passes a constriction, sharp turn, or rough surface. Now, you can really see that here. You can see that this blood flow here, when it kind of narrows down, this tube goes into a constriction, starts that kind of movement. That actually causes vibrations. The chaotic blood flow causes vibrations. That vibration actually creates sound. We listen to that through a blood pressure cuff when we take somebody's blood pressure. So blood pressure can be measured in two ways. A mercury manometer, which is the blood pressure cuff with the stethoscope and the gauge, which is the mercury manometer, and a cannula, which is a direct, like you put an actual tube into a blood vessel. When we are using the blood pressure cuff, we're actually listening to for a noise called a Korotkov sound, okay? This sound right here. When it starts to loosen and we can hear that turbulent flow, that's the sound we're looking for. Now, here's my blood pressure cuff. Here's the example. When you are taking somebody's blood pressure, you make the cuff so tight that literally it squishes that brachial artery closed. Now remember, you've got systolic pressure and you've got diastolic pressure, right? So you start to undo the little dial, and the blood pressure cuff starts getting looser around this person's arm. Now, why is that important? Because when the heart contracts with enough force to actually force that vessel open, that's the systolic sound you're hearing. That's literally, I'm pushing with enough force that I'm opening that vessel for just a second, and you hear that. You hear the Korotkov sound. So that first sound that you hear in a blood pressure cuff is the systolic pressure. Okay? Now, as you keep loosening that valve, eventually you're going to get to a point where there is no Korotkov sound. When that happens, that's when that sound disappears. That's the diastolic pressure. That's when the heart is relaxing. There still is no odd sound that you're hearing. Because as you release that cuff, the systolic is going to open this more and more, but eventually the blood vessel will open up even when the heart is relaxing. But since it's not completely opened up, you still hear the whooshing sounds. When you get to the point where there is no sound anymore, like I said, that blood vessel is open regardless of whether your heart is contracting or relaxing. So that's systolic contracting pressure and diastolic relaxing pressure. Dun, da, da. Okay, Poisson-Yes's law and Laplace's law. So Poisson-Yes's law, a small change in the radius dramatically changes resistance to flow, thus the amount of blood flowing through the vessel. Okay, so if I've got a vessel this big carrying blood and I've got a vessel this big carrying blood, which one carries more blood? Obviously, it's this one because it's bigger. That's all this is saying. So the smaller the vessel, the less blood flowing through that vessel. The smaller the radius, the less blood flows. Now, Laplace's law is a bit different. The force acting on the wall of the blood vessel is proportional to the diameter. So if I have a blood vessel this big, the amount of push along the walls of this vessel is less than if I have a blood vessel this big, because this is so much smaller, the amount of blood pushes harder up against the walls. That's all Laplace's law is saying, and what Poisson-Yes's law is saying. So here's the pronunciation for Poisson-Yes's law, if you want it. I am not French, so I'm doing the best I can. Blood flow is the amount of blood flowing through a vessel in a given period. I don't think that's a big 411. You increase blood pressure, you increase the push behind it, you increase the amount of blood flow. If I turn on a water spigot harder, the amount of water flowing through that hose is more. You increase resistance, you decrease blood flow. So basically, a clean pipe moves more water than a dirty pipe. That's all that's saying. Resistance. What can cause resistance? Well, how thick is your blood? If your blood is very, very thick, it's not going to flow very well. Water versus pancake syrup. Pancake syrup doesn't really flow very well at all. Changes in blood vessel diameter. A blood vessel this big will move more than a blood vessel that big. The length of a blood vessel. So think about throwing a ball. The further that the ball gets away from where you originally threw it, the less likely it is to go further. So I can throw a ball as hard as I can, and maybe I'll say, let's go with my pipe dream, I can get it to fly for 30 yards. But as it goes that 30 yards, if nothing gets in the way, nobody catches it, it kind of starts to lose momentum, and eventually it'll hit the ground, right? The longer the blood vessel is, the harder it is for it to keep going. I'm never going to be able to throw a ball for 100 yards. There's no way. I just don't have enough power behind it. I hope that makes sense. Okay, viscosity is the thickness. How resistant is it to flow? Pancake syrup versus water. And for our body, how thick our blood is dependent on the concentration of red blood cells. How many red blood cells are there? The more there, the more thick. We already said this in that last slide, so I'm not going over it again. We already did this, so I'm not going to go over that again. So as pressure in a vessel decreases, the force holding the vessel open decreases, and the vessel tends to collapse. So you've got something called critical closing pressure. Think about a water balloon, or any balloon for that matter. If you have air in there and there's pressure inside of it, it stays inflated. But the minute that you let the air out, it collapses. The pressure is what's keeping that balloon, the sides of that balloon, separate from each other. Your arteries and your veins, to a certain extent, are kind of the same way. You have to have fluid in those veins or those vessels in order for them to stay open. When that pressure isn't enough, they'll deflate just like that balloon will. That kind of all or none moment where they're going to deflate because there isn't enough pressure inside to keep them open is the critical closing pressure. Okay, compliance is the tendency for a blood vessel volume to increase as blood pressure increases. Remember that our blood vessels will expand if you put enough pressure behind them. So compliance is the tendency for the volume to get higher as the pressure gets higher, as they stretch out to accommodate that pressure. Vessels with high compliance exhibit large increases in volume when pressure increases a small amount. And vessels with low compliance aren't as stretchy, meaning that I can increase pressure, but the volume won't expand anymore. Veins. If you think back to lab, arteries were really, really thick walled. They don't have a big compliance, whereas veins look like somebody has a deflated balloon somewhere. They have 24 times greater compliance than arteries. They are able to stretch to accommodate changes in pressure quite a bit more than arteries. And what that means is I can actually use veins as storage areas because they can hold larger volumes. Okay. Most of our blood resides in our body because we have more arteries and veins there. But out of that, the majority is sitting in our veins, which we just said 2 slides ago, that they act as storage areas. So that makes sense. I'm not going to worry about this, so don't worry about this slide. As blood pressure goes from immediately leaving the heart at the aorta all the way until it reaches the right atrium, it's going downhill. It's going from a high pressure to a low pressure. So literally, this is kind of like diffusion. You've got high pressure as it goes into the aorta, and then it hits zero when it gets to the right atrium. So movement is actually proportional to resistance. And that's all I really want you to know off that slide. Pulse pressure. We all know that the radial pressure, the radial pulse on the wrist is where we usually take somebody's pulse. Pulse pressure waves are actually traveling through the vascular system faster than the blood flows because we're measuring that force. We're not actually measuring the flow of the blood. You have different places on the body that you can actually take somebody's pulse. If there's a leg crush injury somewhere up here, a lot of times doctors will feel these 2 places to see if there's blood flow going down there, to see if they have to compensate for tissue death or if the tissue has been dead for so long that nothing they do will help. Okay, capillary exchange and regulation of interstitial fluid volume. 3 things affect the movement of fluid from the capillaries. So you have to remember that the capillaries are our smallest vessels where we have exchange between the tissues and the blood. Okay? So 3 things affect this exchange, whether things are moving in or out. The blood pressure, the capillary permeability, how holy are those capillaries, and osmosis, because remember, things dissolve in water, so having water moving back and forth kind of helps us to move things. Capillary exchange is the movement of substances into or out of the capillary. It's the most important method used, or I guess I should say the most important method used is diffusion. Again, movement from a high concentration to a low concentration where it doesn't cost us any energy. Nutrients and gases and hormones will move this way. They sometimes, depending on where they are, they can go through fenestrations. Fenestrations are a very fancy way of saying windows. So in some capillaries, we actually have these small, teeny tiny pores like this called fenestrae. which just like I said means windows. Depending on where you are, we kind of talked about this when we talked about water-soluble hormones and how we have to have windows for them to get in and out of. There is some pinocytosis. Pinocytosis, remember, is cell drinking, but not much, to be honest, not really a lot. Net movement occurs from the blood vessel into the tissues. So net movement is going from in here to out here into the tissues, from in the blood vessel out into the tissues. And because we do tend to lose fluid because of things like fenestre, we do use the lymphatic system to pick up any excess.